No man's an island. Well, the same goes for your cells too. So, picture this. Your messenger molecules are like the partygoers, the receptors are the bouncers, the club being the cell. So, welcome to the wacky world of intracellular signaling. Cells communicate with each other through extracellular molecules. The extracellular messenger molecules can either be hydrophobic or hydrophilic. The hydrophilic are easily transported, dissolved in plasma. The hydrophobic need to be carried by plasma proteins to their effector cells, much like they need a cab. The hydrophobic are also lipophilic. So they can simply diffuse through the lipid plasma membrane and directly interact with your nuclear receptor. The focus of this video is on the hydrophilic molecules, which are also lipophobic, and they need a cell surface receptor to transmit their signal to the interior of the cell. Now, based on the distance these signals travel, these messenger molecules or the signaling is classified as autocrine signaling, where the cell that is producing the extracellular messenger will express the receptor on its own surface and respond to it. Secondly, we have paracrine signaling, where the messenger molecule has to travel only a short distance through the extracellular space to the cells that are in close proximity to the cell that is generating the messenger molecule. Now, the paracrine messenger molecules are either unstable, degraded by enzymes, or they bind to the extracellular matrix. These can be the probable reasons why they are unable to travel around the body. Then we have endocrine signaling, where the messenger molecules reach their target cells via passage through bloodstream. Lastly, there is also direct signaling, when the two cells bind to each other because they express complementary proteins on their surfaces. And moment the cells bind to each other, there is a conformational change either in one or both the cells, resulting in transmission of the signal. Now, let's familiarize ourselves with certain terms. You already understand what we mean by receptor. The name given to the extracellular messenger molecules is ligand. Now, the cell will only respond to a particular ligand if it is expressing a receptor that will recognize it specifically. So, no receptor to the ligand, no response. Now, you will assume that if the cell has the same receptor and same ligand, the response is same. It is not the case. Cells that share a specific receptor may respond very differently to the same extracellular ligand. For example, both liver cells and smooth muscle cells express beta-2 adrenergic receptor. Now, this receptor on activation with adrenaline will lead to breakdown of glycogen in the liver cell and relaxation of the smooth muscle. The reason for the different outcome is traced to the different intracellular proteins that become engaged in response in the two types of cells. In a nutshell, signal transduction means conversion of the extracellular signal to intracellular signal. The extracellular messenger molecule now must bind to the receptor on the outer surface of the responding cell. And this interaction actually induces a conformational change in the receptor that causes the signal to be relayed across the plasma membrane to the cytoplasmic domain. And once this signal has reached the inner surface of the plasma membrane, there can be two major routes by which the signal is transmitted in the cell interior. One type of receptor will transmit the signal from its cytoplasmic domain to a nearby enzyme. This enzyme generates a second messenger. Second messengers are small substances that are specific proteins, and depending on their chemical structure, the second messengers can diffuse through the cytoplasm or remain attached to the lipid bilayer of the membrane. The second type of receptor or the second route involves transforming the cytoplasmic domain into a recruiting station for cellular signaling proteins. And these proteins will interact with one another or with components of cell membrane, resulting in a cascading reaction. The outcome is same in both the position. The protein at the top of the intracellular pathway is activated. Now, these signaling pathways are like information superhighways of the cell. Each signaling pathway will consist of a series of distinct proteins that operate in a specific sequence. And each protein will act in such a way that it will alter the conformation of the subsequent protein in the series. They can either activate or inhibit the protein. Alterations in the conformation are accomplished by two kinds of enzymes, protein kinases and protein phosphatases, that respectively add or remove phosphate group from other proteins. Now, the response initiated by the target cell can either result in a change in gene expression, alteration in the activity of metabolic enzyme, reconfiguration of cytoskeleton, increase or decrease in cell mobility, a change in ion permeability, activation of DNA synthesis, and even apoptosis. I hope the video was helpful. Please like, subscribe, and share.